137 June 2024 exam. 1. A. Fill in the blanks. 1 times 3 equals 3 marks. I. Cell-to-cell movement of water during symplastic pathway takes place across plasmodesmata. E. Conversion of to in eukaryotic cells is mediated by an enzyme nitrate reductase. E. A vena curvature test was proposed by F.W. Wendt. 1. B. Choose the correct option. A. Half times 4 equals 2 marks. I. The enzymes where the site of synthesis is different from the site of action is called exoenzymes. E. Phototropic movement of curvature is an example of induced plant movement. E. Mineral nutrient nickel is a constituent of the enzyme urease. IV. Shade-loving plants are termed scheophytes. 1. C. Define the following terms. 1 times 5 equals 5 marks. I. Imbibition. Imbibition is the process by which living or dead plant cells absorb water by surface attraction without forming a solution. It typically occurs in hydrophilic substances such as cellulose and proteins, leading to swelling. E. Absorption spectrum. The absorption spectrum is a graph showing the specific wavelengths of light absorbed by a pigment. In photosynthesis, pigments like chlorophyll absorb blue and red light most efficiently, as evident from their absorption spectra. E. Chemosynthesis. Chemosynthesis is the process by which certain organisms synthesize carbohydrates by using energy derived from the oxidation of inorganic molecules, e.g., hydrogen sulfide or ammonia, rather than from sunlight, unlike photosynthesis. IV. Vernalization. Vernalization is the process of inducing flowering in plants by exposing them to a prolonged period of low temperatures. It is crucial in temperate plants where cold is required to break dormancy and promote floral development. V. Respiratory quotient RQ. Respiratory quotient is the ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed during cellular respiration. For carbohydrates, the RQ is 1, for fats, it's less than 1, and for proteins, around 0.8. Answer to question 2. A. Composition of photosynthetic apparatus in chloroplast. The photosynthetic apparatus in the chloroplast is a highly organized and complex system that is responsible for capturing light energy and converting it into chemical energy during photosynthesis. The chloroplast itself is a double-membraned organelle found predominantly in the mesophyll cells of plant leaves. Inside the chloroplast, the main structures responsible for photosynthesis are the thylakoids, which are flattened, disc-shaped sacs. These thylakoids are stacked in some regions to form grana, and the fluid matrix surrounding them is called the stroma. Embedded in the thylakoid membranes are protein pigment complexes that form the photosystems. There are 2 main photosystems, photosystem I (PSI) and photosystem II (PSII). These photosystems contain chlorophyll A, chlorophyll B, carotenoids, and other accessory pigments arranged in light-harvesting complexes LHCs which absorb and funnel light energy to the reaction centers. The reaction center of PSII contains P680 chlorophyll A molecules, while PSI has P700 chlorophyll A molecules. When excited by photons, these centers lose electrons that are transferred along the electron transport chain. Key components of the photosynthetic electron transport chain include plastoquinone PQ, the cytochrome B6F complex, plastocyanin PC, ferredoxin FD, and NADP plus reductase. ATP synthase complexes are also embedded in the thylakoid membrane and use the proton gradient generated during electron transport to produce ATP from ADP and inorganic phosphate. The entire system works synergistically, where PSII captures photons and initiates the splitting of water molecules photolysis releasing electrons, protons, and oxygen. The electrons pass through the electron transport chain to PSI, and their energy is used to pump protons into the thylakoid lumen, generating a proton gradient. PSI then transfers the electrons to NADP+, reducing it to NADPH. Both ATP and NADPH are utilized in the Calvin cycle in the stroma to fix carbon dioxide into glucose. B. Photorespiration and strategies to reduce it. Photorespiration is a light-dependent process that occurs in plants where the enzyme rubisco-oxygenates RUBP ribulose-1, 5-bisphosphate instead of carboxylating it, leading to the production of glycolate, a 2-carbon molecule, and the release of carbon dioxide. This occurs when the concentration of oxygen is high and carbon dioxide is low inside the leaf, often under conditions of high temperature, intense light, and drought when stomata are closed to reduce water loss. The process of photorespiration is considered wasteful because it leads to the loss of previously fixed carbon, consumes ATP, and does not produce any ATP or NADPH in return. Photorespiration primarily occurs in C3 plants due to the dual affinity of rubisco for both CO and O. To reduce the effects of photorespiration, plants have evolved several strategies. One of the most important adaptations is the development of C4 photosynthesis. In C4 plants like maize and sugarcane, carbon fixation is spatially separated. CO is initially fixed in mesophyll cells by the enzyme PEP carboxylase into a 4-carbon compound oxaloacetate, which is then transported to bundle sheath cells where CO is released and refixed by rubisco. This mechanism effectively concentrates CO around rubisco, thereby minimizing oxygenation reactions. Another adaptation is found in CAM crassylation acid metabolism plants, such as cacti and succulents. These plants temporarily separate CO fixation and the Calvin cycle. They open their stomata at night to fix CO into malate, which is stored in vacuoles. During the day, the stomata close, and CO is released from malate for use in the Calvin cycle. Both strategies increase the efficiency of photosynthesis under stress conditions and reduce carbon loss via photorespiration. Other approaches to reduce photorespiration include increasing the internal CO concentration by enhancing carbonic anhydrase activity or genetically modifying rubisco to increase its specificity for CO over O, though this is challenging. In summary, photorespiration is a significant limitation to photosynthetic efficiency in many plants, but through both natural adaptations and biotechnological efforts, strategies are being developed to mitigate its effects and improve crop productivity. Answer to question 3. A. Role of phloem elements during loading and unloading. The phloem is a complex tissue responsible for the translocation of organic solutes, especially sugars like sucrose, from source to sink tissues. The major phloem elements involved in this process include sieve tube elements, companion cells, phloem parenchyma, and fibers. The sieve tube elements are elongated, living cells that are arranged end-to-end to form long sieve tubes. These cells lack a nucleus and major organelles to facilitate easy flow of phloem sap but are assisted metabolically by adjacent companion cells, which are connected to sieve tubes by numerous plasmodesmata. During phloem loading, sugars synthesized in mesophyll cells are actively transported into the sieve tube elements via companion cells. This loading process can be symplastic, where solutes move through plasmodesmata, or apoplastic, where they move across cell membranes using active transport proteins. In many plants, the apoplastic pathway is predominant and requires ATP-dependent sucrose transporters. The accumulation of sugars in the sieve tubes lowers the water potential, causing water to enter osmotically from adjacent xylem vessels. This influx generates a high turgor pressure in source regions. As a result of this pressure, phloem sap is pushed toward the sink tissues through mass flow. At the sink, such as roots, young leaves are developing. Fruits, unloading occurs. The sugars are transported out of the sieve tubes into sink cells, again via symplastic or apoplastic pathways. Active transport is often required, particularly when unloading into storage tissues. As the sugars are removed, the osmotic potential in the sieve tube increases, water exits into the xylem, and the turgor pressure is reduced, maintaining a pressure gradient from source to sink. Companion cells play a crucial role in both loading and unloading by regulating solute movement and maintaining the energy supply required for active transport. Phloem parenchyma assists in storage and lateral transfer of solutes, while phloem fibers provide mechanical support. The dynamic and highly coordinated activity of these elements ensures efficient long-distance transport and distribution of nutrients throughout the plant. B. Composition of phloem exudates and role of aphids. Phloem exudates are the fluid contents of the sieve tubes that are rich in a variety of substances. The most abundant component is sucrose, which serves as the primary transported sugar. In addition to sucrose, phloem sap contains amino acids, organic acids, hormones such as auxins, cytokinins, abscisic acid, and gibberellins, as well as signaling molecules like RNAs and proteins. Ions such as potassium, calcium, magnesium, and phosphate are also present. Trace elements, defense-related metabolites like alkaloids and phenolics, and other small molecules may also be found depending on the plant species and physiological conditions. Technically challenging due to the difficulty in accessing sieve tube contents without causing wounding responses that seal the sieve plates. This is where aphids become extremely valuable. Aphids are phloem-feeding insects that insert their stylets into the sieve tubes to feed on sap. Remarkably, aphids can tap into the sieve elements without triggering a defense response or causing significant blockage of the sieve plates, allowing a steady flow of phloem sap through their stylets. Scientists have utilized this by decapitating feeding aphids and collecting the exuding sap from the remaining stylets, which continues to flow due to pressure in the sieve tubes. This method allows researchers to obtain relatively pure samples of phloem exudates and study their composition. Through such studies, the role of phloem is not just a conduit for sugars but also a signaling pathway for systemic acquired resistance, development, and response to environmental stimuli has been revealed. Moreover, analysis of phloem exudates has expanded our understanding of how plants allocate resources, respond to stress, and coordinate growth processes. In conclusion, the cooperation of phloem elements in loading and unloading and the analysis of phloem contents using aphids provide deep insight into the fundamental processes that sustain plant life and productivity. Answer to question 4. Physiological roles and deficiency symptoms of 4 macronutrients. Macronutrients are essential mineral elements required in large quantities by plants to complete their life cycle. Among them, nitrogen, phosphorus, potassium, and calcium play particularly critical roles in various physiological and biochemical processes. Nitrogen is a key component of amino acids, the building blocks of proteins, and is also found in nucleic acids such as DNA and RNA. It forms part of the chlorophyll molecule, enabling photosynthesis. It is also a constituent of ATP and several coenzymes. Physiologically, nitrogen promotes vegetative growth, including leaf and stem development. Its deficiency is characterized by chlorosis or yellowing of older leaves, stunted growth, and poor development. In extreme cases, nitrogen deficiency can lead to complete cessation of growth and premature senescence of leaves, drastically affecting the plant's yield and survival. Phosphorus is another vital macronutrient involved in energy transfer reactions within the plant. It is a component of ATP, NADP, and nucleic acids, making it indispensable for photosynthesis, respiration, and all cellular metabolism. Phosphorus plays a critical role in root development and seed formation. A deficiency in phosphorus causes stunted growth, dark green or purplish pigmentation in older leaves due to accumulation of anthocyanins, delayed flowering, and poor fruit or seed production. Often, the root system remains underdeveloped, which affects the plant's ability to absorb water and nutrients from the soil. Potassium, though not part of any structural molecule, is crucial for osmoregulation, enzyme activation, and maintaining ionic balance in cells. It helps in stomatal movement by regulating guard cell turgor, thereby affecting transpiration and gas exchange. Potassium activates over 60 enzymes involved in processes such as protein synthesis, carbohydrate metabolism, and nitrogen assimilation. Deficiency symptoms of potassium include marginal chlorosis progressing to necrosis on older leaves, weak stems, lodging in cereals, and reduced resistance to drought, pests, and diseases. The inability to maintain turgor pressure and proper enzyme function leads to overall metabolic imbalance and poor crop quality. Calcium is essential for maintaining structural integrity of cell walls, particularly as a component of calcium pectate in the middle lamella. It also plays an important role in membrane stability, cell division, and acts as a secondary messenger in many signal transduction pathways. Unlike other nutrients, calcium is relatively immobile in plants, so its deficiency symptoms first appear in younger tissues. Common symptoms include tip burn in leaves, blossom End rot in fruits like tomatoes, necrosis of meristematic regions, and poor root development. Calcium deficiency severely impacts cell division and expansion, leading to malformed or aborted organs. The uptake and movement of these macronutrients are tightly regulated in plants to maintain homeostasis. Environmental factors such as soil pH, moisture, microbial activity, and temperature can greatly influence their availability. To combat deficiencies, agricultural practices such as balanced fertilization, use of biofertilizers, and soil amendment strategies are employed. For example, legumes can fix atmospheric nitrogen through symbiotic relationships with rhizobium bacteria, improving nitrogen content in the soil. Likewise, adding compost or rock phosphate enhances phosphorus availability. The interplay among these macronutrients is also crucial. For instance, an excess of nitrogen can inhibit potassium uptake, and calcium antagonizes magnesium absorption. Thus, understanding the physiological roles and deficiency symptoms of macronutrients is essential for managing plant health and ensuring optimal growth, productivity, and quality. Furthermore, modern research continues to explore the signaling functions of these elements, particularly potassium and calcium, in stress tolerance, gene regulation, and metabolic adaptation. Ultimately, the balance and bioavailability of macronutrients underpin the entire framework of plant physiology and metabolic activity, reflecting directly on food security and agricultural sustainability. Answer to question 5. Glycolysis pathway with appropriate schematic diagram. Glycolysis is a central metabolic pathway in both plants and animals that involves the enzymatic breakdown of glucose, a 6-carbon sugar, into 2 molecules of pyruvate, a 3-carbon compound, releasing energy in the form of ATP and NADH. This process occurs in the cytoplasm of the cell and is the first stage of cellular respiration, regardless of whether oxygen is present. Glycolysis is divided into 2 phases, the energy investment phase and the energy payoff phase. In the energy investment phase, glucose is phosphorylated and rearranged to form 2 molecules of glyceraldehyde 3-phosphate G3P, consuming 2 molecules of ATP in the process. Specifically, glucose is first converted to glucose 6-phosphate by the enzyme hexokinase using 1 molecule of ATP. Glucose 6-phosphate is then isomerized to fructose 6-phosphate by phosphoglucose isomerase. A second phosphorylation, catalyzed by PHOS-PHOFRUCTOKINASE1, converts fructose 6-phosphate to fructose -1, 6-bisphosphate using another ATP. This molecule is then split into 2 3-carbon sugars, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, by aldolase. The enzyme triose-phosphate isomerase then rapidly interconverts dihydroxyacetone phosphate to another molecule of glyceraldehyde 3-phosphate so that both can proceed through the second phase. In the energy payoff phase, each G3P molecule undergoes a series of reactions that ultimately yield pyruvate. These reactions include oxidation, substrate-level phosphorylation, and rearrangements. Glyceraldehyde 3-phosphate is oxidized by glyceraldehyde 3-phosphate dehydrogenase to form 1,3-bisphosphoglycerate 1,3-BPG, reducing NAD+ to NADH in the process. This step is crucial as it traps energy in the form of NADH. Next, 1,3-BPG donates a high-energy phosphate to ADP, forming ATP and 3-phosphoglycerate 3-PG via the enzyme phosphoglycerate kinase. This is the first instance of substrate-level phosphorylation. The 3-PG is then converted into 2-phosphoglycerate 2-PG by phosphoglycerate mutase, followed by the removal of a water molecule by enolase to form phosphoenol pyruvate PEP. Finally, PEP donates its phosphate group to ADP via pyruvate kinase, yielding a second ATP molecule and pyruvate. Since 2 molecules of G3P are processed per glucose molecule, the energy output is doubled. Thus, glycolysis produces a net gain of 2 ATP molecules and 2 NADH molecules per molecule of glucose metabolized, along with 2 molecules of pyruvate. Here is a textual schematic diagram representation of the glycolysis pathway. peptides, and fatty acids via its intermediates. Additionally, in plants, glycolysis is crucial not just for energy production but also for generating the intermediates required for anabolic processes in actively growing tissues, such as meristems and developing seeds. Because glycolysis does not require oxygen, it allows energy production to continue under hypoxic or anaerobic conditions, such as waterlogged soils in plants. The pathway is tightly regulated at key enzymatic steps, hexokinase, phosphofructokinase, and pyruvate kinase, to ensure cellular energy balance and adapt to metabolic needs. In summary, glycolysis is a vital, universal pathway central to energy production and metabolic integration in all living organisms. Answer to question 6. A. Physiological functions of ethylene in plant growth and development. Ethylene is a simple gaseous plant hormone with profound and wide-ranging effects on plant physiology. Despite its simple structure as a hydrocarbon, ethylene plays key roles in almost every phase of a plant's life cycle, from seed germination to fruit ripening and senescence. It is synthesized in almost all plant tissues but especially accumulates in actively growing regions and in response to environmental stress. One of the most well-known roles of ethylene is in fruit ripening. In climacteric fruits such as bananas, tomatoes, and apples, ethylene triggers a surge in respiration and activates enzymes like pectinases and cellulases that soften fruit tissue, and amylases that convert starch into sugars, leading to characteristic taste, color, and texture changes. Ethylene also regulates leaf abscission and senescence by influencing gene expression and enzyme activity that degrade cell walls at the abscission zone, allowing leaves or fruit to detach. This is particularly important in seasonal leaf shedding in deciduous trees. In seedling development, ethylene mediates the triple response, a morphological adaptation to soil impedance. When seedlings encounter obstacles while growing underground, ethylene causes radial swelling of the hypocotyl, inhibition of stem elongation, and the formation of an exaggerated apical hook. This adaptation helps protect the delicate apical meristem during emergence. Ethylene also plays a crucial role in stress responses. It is produced in greater quantities in response to mechanical stress, pathogen attack, drought, flooding, or ozone exposure. In such scenarios, it modulates the expression of stress-related genes and coordinates with other hormones like abscisic acid and salicylic acid to activate defense mechanisms. Moreover, ethylene influences flowering in certain species, especially bromeliads and pineapples, where its application can induce synchronized flowering. In root development, ethylene interacts with auxin to promote lateral root formation and adventitious root initiation. In high concentrations, ethylene may inhibit root elongation but promote root hair development, enhancing the root's surface area for nutrient uptake. Ethylene's action is mediated by a family of receptors located in the endoplasmic reticulum membrane. Upon binding ethylene, these receptors deactivate a negative regulator, allowing downstream signaling cascades that modify gene expression. One key transcription factor activated in this pathway is EIN3, ethylene insensitive 3, which induces genes that drive the ethylene response. Ethylene also influences sex expression in some monoecious and dioecious plants. For example, in cucumbers, high ethylene levels promote the development of female flowers, a trait exploited in agriculture to enhance fruit yields. Ethylene is also used commercially to synchronize ripening and degreening of fruits like citrus by removing chlorophyll. However, excess ethylene in storage facilities can lead to undesirable over-ripening or spoilage, so ethylene absorbers or inhibitors are often used in post-harvest technology. Altogether, ethylene functions as a key regulator of plant development, environmental adaptation, and reproduction. B. Phytochrome-mediated response in plants. Phytochromes are specialized photoreceptors in plants that detect red R and far-red FR light and regulate a variety of light-dependent developmental processes, collectively known as photomorphogenesis. These are dimeric proteins consisting of a protein moiety and a light-absorbing pigment called phytochromobilin. Phytochrome exists in two interconvertible forms, the red light-absorbing PR form and the far-red light-absorbing PFR form. Red light, around 660 nanometers, converts PR to PFR, the biologically active form, while far red light around 730 nanometers converts PFR back to pastel. This photoreversibility forms the molecular basis of light perception and signaling in plants. Upon activation by red light, PFR migrates into the nucleus and interacts with specific transcription factors such as PIFs, phytochrome interacting factors, leading to the activation or repression of light-responsive genes. Phytochrome-mediated responses are broadly categorized as very low fluence responses (VLFR), low fluence responses (LFR), and high radiance responses (HIR). Examples of phytochrome-regulated processes include seed germination, stem elongation, leaf expansion, flowering, and shade avoidance. In Arabidopsis and lettuce, phytochrome activation by red light promotes seed germination, while far red light inhibits it. This is particularly important in buried seeds that use red-far red light ratio as a cue for proximity to the soil surface. In the shade avoidance response, plants perceive a low red to far red light ratio under canopy conditions, triggering elongation of stems and petioles to outgrow competitors. This is mediated by decreased PFR levels and increased expression of genes associated with cell elongation. Phytochrome also plays a role in circadian rhythms and flowering time. In photoperiodic flowering plants, phytochrome helps measure day length. In long-day plants, the presence of PFR at the end of the light period promotes flowering, while in short-day plants, its presence may inhibit flowering. Phytochromes regulate the expression of constants and flowering locus T genes, which are central to the floral induction pathway. Phytochrome A and B are the most studied among several isoforms. Phi A primarily mediates responses under far red and continuous light, while Phi B predominantly senses red light. In daytilation, phytochromes suppress scotomorphogenesis, growth in darkness, causing the inhibition of hypocotyl elongation and the opening and greening of cotyledons when the seedling is exposed to light. Mutants lacking functional phytochrome genes exhibit altered photomorphogenic development, demonstrating their crucial role in plant light signaling networks. In conclusion, ethylene and phytochrome are two significant regulators in plants. Ethylene is a chemical signal modulating growth and stress responses, and phytochrome is a light sensor directing developmental transitions and environmental adaptability. Answer to question 7. 8 factors that affect the rate of enzyme activity. Enzymes are biological catalysts that accelerate chemical reactions in living organisms, and the rate at which they act is influenced by a variety of physical, chemical, and biological factors. One of the most critical factors affecting enzyme activity is temperature. Each enzyme has an optimum temperature at which its activity is maximal. In plants, this is generally around 25 to 35 degrees Celsius, although it varies with species and cellular location. When the temperature is too low, the kinetic energy of molecules is reduced, slowing down molecular collisions and the frequency of enzyme-substrate complex formation. As the temperature increases, the rate of enzymatic reactions also increases up to the optimal point. However, beyond the optimum, enzymes begin to denature as the high thermal energy disrupts the hydrogen bonds and other interactions, maintaining the enzyme's tertiary structure, leading to a decline or total loss in catalytic activity. pH is another significant determinant of enzyme activity. Each enzyme functions best at a specific pH, which is usually related to the environment in which it operates. For example, cytosolic enzymes often have an optimum pH near neutral, while those in vacuoles or lysosomes may work best under acidic conditions. Deviations from the optimal pH alter the ionization of amino acid residues, particularly those at the active site, and can disrupt enzyme-substrate interactions or cause denaturation. Substrate concentration also directly influences enzyme activity. As substrate concentration increases, the rate of reaction increases due to more frequent enzyme-substrate collisions. This relationship follows Michaelis-Menten kinetics. Initially, the reaction rate rises sharply with increasing substrate, but eventually reaches a plateau where all enzyme active sites are saturated. At this saturation point, increasing substrate concentration further will not increase the reaction rate, as the enzyme becomes the limiting factor. Enzyme concentration also determines the reaction rate, provided substrate is in excess. Higher Concentrations of enzyme allow for more substrate molecules to be processed simultaneously, leading to increased reaction rates. However, this only holds true as long as other factors like temperature and pH remain optimal. The presence of cofactors and coenzymes also affects enzyme activity. Many enzymes require non-protein helpers such as metal ions, e.g., Mg², Fe², Zn², or organic molecules like NAD or FAD to function properly. These molecules may be tightly or loosely bound to the enzyme and are often essential for catalytic activity. A deficiency in necessary cofactors can significantly reduce enzyme function. Inhibitors also have a strong impact on enzyme activity. Competitive inhibitors resemble the substrate and bind to the active site, preventing substrate access. This type of inhibition can often be reversed by increasing substrate concentration. Non-competitive inhibitors bind to sites other than the active site, altering the enzyme's shape and reducing its activity regardless of substrate concentration. Incompetitive inhibitors bind only to the enzyme-substrate complex, and all forms of inhibition reduce overall enzymatic efficiency. Allosteric regulation also plays a vital role in modulating enzyme activity. Allosteric enzymes possess multiple binding sites, and binding of effectors, activators or inhibitors to allosteric sites induces conformational changes that either enhance or reduce enzyme activity. This type of regulation is common in key metabolic enzymes, allowing cells to fine-tune pathways in response to changing needs. Post-translational modifications like phosphorylation, methylation, and ubiquitination can also influence enzyme activity by changing their structure, stability, or localization. Environmental stress, such as salinity, drought, or heavy metal toxicity, can also modulate enzyme activity by altering the cellular ionic balance, damaging enzyme structure, or modifying gene expression patterns. Finally, enzyme activity can be regulated genetically at the level of transcription and translation. Genes encoding enzymes may be upregulated or downregulated in response to internal signals like hormones or external stimuli such as light or pathogen attack. This multi-layered regulation ensures that enzymes are synthesized and activated precisely when and where needed. B. Concept of holoenzyme. A holoenzyme refers to the complete, catalytically active form of an enzyme, consisting of the apoenzyme and its necessary cofactors or coenzymes. The apoenzyme is the protein portion of the enzyme, which by itself is inactive. The non-protein part can be a metal ion or an organic molecule, and when it binds to the apoenzyme, the entire structure becomes functional. For example, in RNA polymerase, the core enzyme requires a sigma factor to form the holoenzyme capable of initiating transcription. In plant systems, several holoenzymes like nitrate reductase require molybdenum cofactor for activity. Holoenzymes play essential roles in metabolism and signal transduction by integrating structural and functional elements into a complete, responsive catalytic unit. The formation of holoenzymes ensures specificity, proper orientation of substrate, and stabilization of the transition state, making enzymatic reactions efficient and highly regulated within the cellular environment. Answer to question 8. I. Active transport. Active transport is the energy-dependent process by which molecules and ions are moved across biological membranes against their concentration gradient, from regions of lower concentration to regions of higher concentration. Unlike passive transport, which relies on diffusion and does not require energy, active transport requires the direct or indirect expenditure of metabolic energy, usually in the form of adenosine triphosphate (ATP). In plants, active transport is crucial for the uptake of essential minerals and nutrients from the soil, where these substances may be in lower concentrations than inside the root cells. It also maintains ion gradients across membranes, which are necessary for processes such as osmoregulation, turgor maintenance, and the generation of membrane potentials. Transport proteins such as proton pumps, sodium-potassium pumps, and calcium ATPases are embedded in the cell membrane and function as molecular machines that carry out active transport. Primary active transport uses ATP directly, while secondary active transport relies on the electrochemical gradient established by primary transporters. For example, the proton pump HATPase in plant root cells expels protons into the soil, creating a proton gradient that drives the uptake of nutrients like nitrate or phosphate through co-transporters. This mechanism not only aids nutrient uptake but also regulates cytoplasmic pH and supports other physiological processes such as stomatal opening and phloem loading. Active transport systems are highly selective and regulated, ensuring that specific substances are transported only when needed. The disruption of active transport can result in nutrient deficiencies, ion imbalances, and impaired growth, highlighting its vital role in maintaining plant homeostasis and productivity. Role of heat shock proteins in stress tolerance. Heat shock proteins HSPs are a group of highly conserved proteins that are rapidly synthesized in response to elevated temperatures and other forms of stress, such as drought, salinity, heavy metals, and oxidative damage. They act as molecular chaperones that stabilize nascent polypeptides, assist in the proper folding and refolding of denatured proteins, and prevent protein aggregation under stress conditions. The accumulation of HSPs is a universal protective mechanism seen in almost all organisms. In plants, HSPs play an essential role in ensuring cellular survival under abiotic stress by maintaining protein homeostasis, or proteostasis. Various classes of HSPs are categorized based on their molecular weights, such as HSP 100, HSP 90, HSP 70, HSP 60, and small HSPs. HSP 70, for instance, is known for its role in protein folding, while HSP 90 is involved in stabilizing signaling proteins and receptors. The expression of HSPs is regulated by heat shock transcription factors. HSFs, which become active during stress and bind to heat shock elements in the promoter regions of HSP genes. This rapid response helps the cell cope with sudden protein denaturation and cellular damage. Beyond thermal stress, HSPs have been implicated in pathogen resistance and developmental regulation. They are also used as biomarkers in breeding stress-tolerant crop varieties and in genetic engineering for enhancing stress resilience in plants. By maintaining the integrity of the proteome, HSPs contribute significantly to stress tolerance and survival under fluctuating environmental conditions. Cryptochromes. Cryptochromes are flavoproteins that function as blue light photoreceptors in plants, animals, and microorganisms. In plants, they are primarily involved in regulating light-mediated growth and development processes, including seedling deethylation, photoperiodic flowering, circadian rhythms, and entrainment of the biological clock. Cryptochromes absorb blue UV light approximately 340 to 500 nm and undergo conformational changes that activate signaling cascades. They are structurally related to DNA photolysis but do not exhibit DNA repair activity. In Arabidopsis thaliana, Cry1 and CRY2 are the two well-characterized cryptochromes. Cry1 is mainly involved in inhibition of hypocodal elongation under blue light, while CRY2 influences flowering time and circadian rhythms. Cryptochromes interact with a range of signaling proteins and transcription factors to regulate gene expression. For example, they can repress the activity of COP1, a central repressor of photomorphogenesis, thereby promoting light-responsive developmental processes. Cryptochromes are essential for fine-tuning plant responses to environmental light cues and optimizing resource allocation for growth. They also integrate light signaling with hormonal pathways, particularly auxins and gibberellins, affecting plant architecture and reproductive development. The manipulation of cryptochrome function holds potential in crop improvement for enhancing light use efficiency and stress tolerance. Chemiosmotic mechanism of ATP synthesis. The chemiosmotic mechanism of ATP synthesis is a fundamental process by which cells generate ATP, the universal energy currency, and has since become a cornerstone of cellular bioenergetics. In plant cells, chemiosmosis occurs primarily in two organelles, chloroplasts during photosynthesis and mitochondria during respiration. In chloroplasts, light energy drives electrons through the photosynthetic electron transport chain in the thylakoid membrane, resulting in the pumping of protons from the stroma into the thylakoid lumen. This creates a proton gradient or proton motive force (PMF) across the membrane. The potential energy stored in this gradient is harnessed by the enzyme ATP synthase, which allows protons to flow back into the stroma. As protons pass through ATP synthase, it catalyzes the phosphorylation of ADP to ATP. This process is known as photophosphorylation. In mitochondria, a similar proton gradient is established by the electron transport chain on the inner mitochondrial membrane during oxidative phosphorylation. Electrons from NADH and FADH reduce oxygen to water, and the energy released drives proton pumping from the matrix into the intermembrane space. Again, ATP synthase uses the PMF to synthesize ATP. The chemiosmotic theory unified understanding of ATP production across biological systems and highlighted the central role of membrane potential in energy metabolism. It emphasizes the importance of intact membranes, properly functioning proton pumps, and the coupling of electron flow to proton translocation. Disruption of this process can lead to impaired ATP synthesis, affecting all energy-dependent cellular processes including biosynthesis, transport, and stress responses.